Okay, today we're going to start on mapping, which is the beginning of the next section of the course, to talk about linkage and two-factor cross basics. Okay, first of all, there's actually different types of maps, and we're mostly going to be discussing recombination or genetic mapping, where the map distance is based on a calculation of recombination frequency. But there are other types of maps also that we've seen, especially the cytological, where we've seen the banding patterns on chromosomes. And finally, there's a physical map, which is based simply on the DNA sequence. And we'll be looking at issues having to do with how the physical map and the genetic map don't necessarily match each other. And it tells us interesting things about the human genome and its organization. Here's a picture that relates those things. The genetic map is shown. The units here are centimorgans, where one centimorgan is 1% recombination. The cytogenetic map, of course, are the pictures we've been seeing, where the genes are located along the chromosomes, of course, but each chromosome has its characteristic banding patterns, which can help you to locate particular genes. And then finally, the physical map in terms of the base pairs, which we'll talk about a lot more later. Okay, so units of distance, genetics, of course, is recombination frequency. It's also called a map unit or a centimorgan, and it's simply measuring in a genetic cross how frequently crossover occurs as an indirect measure of distance. Now, physical mapping, of course, is units of base pairs in kilobase pairs when we're talking about genes, megabase pairs when we're talking about chromosomes. So in terms of mapping, if you're going to try to do a map genetically, you need to have markers, which is to say places where the DNA differs from one chromosome or one individual to another. Now, many times we'll be following phenotypes that result from genetic mutations, which lead to alterations in the appearance of the organism. But equally good, sometimes better as markers, are DNA sequence polymorphisms, including the STR loci, which we've seen before, and the SNPs especially, which is single nucleotide polymorphisms. So any genetic variation of that sort that you can detect can be used to contribute to your map. Okay, so in terms of recombination, which we'll spend most time on, is you're going to be measuring crosses, doing crosses in which heterozygotes are producing progeny, and the relative proportion of gametes containing the parental and recombinant types will determine the map distances. Now, if you think about it, you can only measure recombination in heterozygotes because you need to have phenotypes that you can score. And so we're going to look for rearrangements in the progeny, indicating that recombination has occurred. So first we have to talk about some vocabulary. What is a parental and what is a recombinant in a genetic cross? Okay, so for independent assortment, consider the cross big A, big B, times little a, little b. The big A, big B parent will give all big A, big B gametes, and little a, little b1 will give all little a, little b gametes. But then you make a heterozygote from here, and of course you're now big A, big B, little b. And when those individuals undergo meiosis, all four gamete types will be formed in equal frequency because of independent assortment. So in this situation, the parents are the parentals are the big A, big B, and little b, and those are the ones that were contributed by the dominant parent to the initial heterozygote. The other two gametes are the little a, little b, little b. Those are the other parental, or the little a, little b is the other parental, because those are the ones that were formed in the heterozygote in the first place. So the original parents of the heterozygote in which we are monitoring recombination, the parentals were big A, big B, and little a, little b. So that what that says to us is that in independent assortment, you're going to get half of your gametes are going to be recombinant, the other and half are going to be parental. So 50 map units, 0.25 over 1 is 50 map units, independent assortment, no linkage. Now, there's different ways you can get recombinants, obviously. These are recombinant in the sense that they're different from what the original parentals were, but they didn't arise by recombination within the chromosome. They arose by independent assortment. Nonetheless, the word recombinant is used to describe those offspring. Okay, so also for independently assorting genes, and this is again the parentals idea, we could have made heterozygote by making little a, big B, big A, little b. In this case, the parentals, which are the contributors to the heterozygote, are little a, big B, and big A, little b. The recombinants are little a, little b, and big A, big B. Again, independent assortment gives all four gamete types in equal frequency, 0.25 each. Now, for linked genes, this is a different situation because the genes are located on the same chromosome and they will require a crossover to separate the original arrangements. And that's going to be the basis of our mapping. Okay, so independent assortment, all gametes equally likely. The recombinants occur with equal frequency compared to the parentals. They obey the rules of independent assortment. For linked genes, however, the gametes are not equally likely because it takes a crossover event to rearrange the parentals. And we're going to use those combinations or recombinants to produce chromosome maps of genes. Okay, so intuitively here, for linked genes which are close together on the same chromosome, the proportion of recombinant and parental depends on the distance between the two genes because you need to have a crossover. And that's going to depend on how far apart the genes are. They can be very small, they can be very close and give you very few recombinants, or they can give you very high recombinants in cases where they're far enough on the chromosome that they're actually assorting independently. So here's a picture of crossing over to produce gametes, and we need a slightly different nomenclature here. We have our parentals, the big A, big B, little b, are forming this heterozygote. Now we have this horizontal line which connects the A's and the B's, indicating that those genes are linked. They are located on the same chromosome. And you need that to describe the situation so that you can see what crossing over can occur. Now when a crossover occurs, of course, it's normally within paired homologs in the Pachytene stage of meiosis, and you show all four chromatids, two of which are involved in the exchange, and two of which are not. But when you're considering just recombination frequency, you don't need to put all four chromatids. You just need to look at the ones where the recombination actually happened. So the more convenient way to show this is, again, the big A on the lower left, the big A, big B representing one homolog, the little a, little b representing the other homolog, and the horizontal bar between them indicating the chromosome. And so I've made this little squiggle to diagram what a recombination event would look like, where you have recombinant types forming, big A, little b, big B. Okay, so that's what a single crossover does. It produces two recombinants, which are considered reciprocals of each other, because whenever you get one recombinant, big A, little b, or big A, little b, or little a, big B, it's a result of a single event which produced both of them, because you're exchanging parts on the chromosomes. Okay, intuitively, if you have a chromosome, which I've drawn here, with the line and the genes right next to each other, you're going to produce all four kinds of progeny, but they're not going to be in equal frequency because the genes are close enough that recombination does not occur frequently between them. Okay, by contrast, in this picture, I have the big A, big B separated by a long distance, allowing much more opportunity for recombination. So if the genes are linked but further apart, you're going to have more recombination and more likely to observe recombinant progeny. And so the big A, little b recombinants and little a, big B will be proportionally more. Okay, also notice before we start, the parentals are not necessarily the dominants and the recessives. There's two ways you could put these genes together. You could have big A, big B on one homolog, little a, little b on the other homolog, and this is called the cis configuration. Or else the parentals could be the trans configuration, which is big A, little a, big B. So don't think the dominants and recessives are always parentals, because they're not. Okay, in some cases we know what the parentals are ahead of time. If we make a cross that we know is big A, big B times little a, little b, then you know what the heterozygote configuration is. The parentals are big A, big B, and little b. And if we did it the other way around with big A, big B, big A, little b, times little a, big B, then the parentals are big A, b, little a, big B. Sometimes we don't know this, but we can usually guess it from what the progeny ratios look like. So this is what I just said before. If we know that these are the parentals, we've got little a, big B, and big A, little b, then we write that parental, or we know what the F1 is, and it has a big A, little b on one homolog, and little a, big B on the other homolog. So depending on what the original arrangement is, two of the progeny types are going to be parental, and two are going to be recombinant. Okay, in general, the parentals will be the most prevalent of the progeny, and the recombinants are less prevalent, and they are reciprocals of each other. So we're going to calculate the map distances by adding the recombinants and divide by the total. In general, recombination frequency mapping, the distance, not physical distance, but genetic distance from gene A to gene B, is the sum of the two recombinants divided by the total. And then multiply by 100 to get map units or centimorgans. Okay, so here's an example. Now you can't know what these numbers are going to be ahead of time because it depends on how far the genes are apart. And so this is just an example to show what kind of calculation needs to be done. Okay, so we've got a big A, big B, little b individual. They undergo meiosis, and we're producing the following types of gametes. Big A, little b, 0.45, little a, big B, 0.45, big A, big B, 0.05, little a, little b, 0.05. So we can see very clearly that there are four gamete types, but they're not at equal frequency. But the two types, the recombinants and the parentals, are reciprocals of each other. You can see the 0.05 matches a 0.05, and the 0.45 matches a 0.45. So in terms of calculating the map distance between A and B, it's very simple, really. Okay, so I just drew a picture here showing what's happened, that we've made this cross, and we get the different proportions of the different gametes, and we're going to use that to calculate our map distance. Well, we've got our recombinants are 0.05, which is to say 5% each, plus 0.05, so you add the two recombinants together, and the total is 1, so that works out to 10 centimorgans, or 10 map units apart. An individual heterozygous for A and B form the following gametes. Big A, little b, 40%, little a, big B, 40%, big A, big B, 40%, little a, little b, 10%, rather 10%, little a, little b, 10%. The parentals are, okay, so the answer is B, big A, little b, and little a, big B. Notice that they're reciprocals of each other. You've got one allele of each. You've got one A, one big A and one little a, one big B and one little b. So the answer is B. Okay. An individual heterozygous for A and B form the following gametes. Big A, big, little b, 40%, little a, big B, 40%, big A, big B, 10%, little a, little b, 10%. The map distance between A and B is therefore, okay, so take the 10 and, well, 0.1 and 0.1, add them together and get 0.2, divide by 1, and you get 0.2, which is 20 map units after you multiply by 100. Right? Right. Okay. Individual heterozygous for genes A and B forms gametes, and 15% of them are big A, little b. What proportion of the progeny will be little a, big B? Okay, so the point here is that you know what the reciprocals are of each other, and they're equal in frequency roughly to each other. So we know that if we have a big A, little b, and that's 15%, its reciprocal is little a, big B, because we need one allele of each. So we're going to have 15% of the reciprocal, which is little a, big B. So the answer is A. Right, the answer is A. Okay, So this was a little more, not difficult exactly, but needs to take a little more thought. The individual is heterozygous for A and B, and 15% of them are big A, little b. Same scenario as before. What proportion are expected to be big A, big B? Well, here we've got to do a little more arithmetic. We know already that 15% is big A, little b, so that means that another 15% are going to be little a, big B, like we saw a minute ago. So the sum of those two is 30%, or 0.3. So the rest of them are the parentals, and so we need to subtract from 1 to get the total of the parentals. So we do that by saying, okay, 1 minus 0.3 equals 0.7. But the big A, big B, that's only half of those, because we have another parental, which is little a, little b. So the answer here is C, because we have to account for all 4 progeny types, only half of which are the big A, big B that we're being asked about. So this is the answer. The answer is C to my previous question, that we're going to have 0.35, which is half of 0.7, which is the total parentals. Well, the total parentals is 0.7. Each of the parentals is half of that, which is 0.35. Okay, realistically, we don't count gametes. You can't reasonably do that. Instead, we have to count progeny. So in doing these crosses, what we're always doing is a cross times a homozygous recessive. Okay, this is an important point because you can't count gametes, but you still want to know what happened during meiosis in that heterozygote. And if you've got another parent which has dominant alleles, you're not going to be able to tell what gamete was produced by the heterozygote. Because remember, that's what we're trying to do, is count up recombinant gametes to get our recombination frequency, or genetic distance. So visualizing this, your heterozygote makes 4 gamete types in equal frequency. Your big A, big B, little a, little b, big A, little b, little a, big B. And in order to visualize, identify, look at the actual gamete, what you have to do is make a progeny. And when you cross times the homozygous recessive, you know exactly what alleles were contributed by that homozygous recessive. And we know what the genotype of that progeny is going to be. And therefore, we can say that any dominant alleles must have come from the heterozygous parent. So basically, what this is saying is we can infer the genotype of the gamete by looking at the phenotype of the progeny, because the homozygous recessive testcross parent only contributed with recessive alleles. So if the phenotype is big A, big B, we know the gamete type was big A, big B from the heterozygote. If it's little a, little b, well, we know there were recessive alleles from both parents, implying that the heterozygote gave both recessives. If you have a big A phenotype, little b, same thing, the big A must have come from the heterozygote, and both parents contributed the little b. Finally, the last one, little a, big B, you got the big B that must have come from the heterozygote, and all the other alleles are recessive. So the point is that you can make a cross, measure progeny, and know for sure, infer what was present in the gamete from the heterozygous parent. And that enables us to count progeny to do mapping, not just count gametes, which you can't do anyway. Okay, organism big A, little b, big A, little a, big B, little b is test crossed to little a, little b, and progeny were obtained. What is the full genotype of the phenotypically big A, big B progeny? Okay, so of the choices, you know you're phenotypically big A, big B, but you know that the other 2 alleles came from the recessive parent, so the only reasonable answer here is B. Big A, big B, little b. B. Okay, so we're not really going to be talking necessarily about big A's and big B's all the time. Drosophila melanogaster is a model organism that's been used for genetics for years and years and years. And a lot of the examples we're going to talk about are going to use flies as a model to calculate recombination frequencies and to write crosses generally. Okay, so here's a picture of a fruit fly. It's a model genetic organism, and it has some very obvious phenotypes that you can use and get mutations in order to actually construct genetic maps. Okay, so in terms of the nomenclature, which is more confusing than anything, Genes are named according to the phenotypes that they produce when mutated, which makes sense. The white eye, for example, the gene is needed for normal eye color, which is red. So in the absence of the normal white gene, which is to say there's a mutant in the white gene, the phenotype is white. So the gene or locus is named W because when it's mutated, the phenotype is white eye. Similar idea for something called vestigial wing. That obviously the gene is needed for a normal wing shape. In its absence, the wing is shriveled and small, and the gene was named vestigial. Okay, so the kinds of phenotypes one looks at in flies have to do with some of these very visible morphological changes. You've got ebony body, vestigial wings, curled wings. Many different colors of eye color are very commonly used in terms of were commonly used to make maps. And here's a picture of all different sorts of ones. We've got one with a yellow body, white eye, sepia eye, curled wing, dumpy wing, black body, ruby eye, vestigial wing, etc. So these are the types of morphological markers that were used in constructing the earliest genetic maps of fly and everybody else. Here's a picture of the genetic map well, the old genetic map of fly. And you can see the genes are arrayed along the chromosome. And they're named according to the phenotypes that they produce when mutant. One thing to notice here that's very important, even though 50 map units is what you get if you do recombination, well, if for independent assortment, 50% is the maximum recombination you can measure in a cross. These chromosomes have more than 50 map units. This chromosome has 107, for example. This one has 106. And the reason for that is you could make crosses in pairwise combinations and build up maps from smaller crosses where you wouldn't be able to detect linkage between genes on one end of this chromosome and genes on the other unless you had intervening genes that you could say they're both linked to those. So don't think that 50% recombination means all chromosomes are 50 map units long, but also maybe more importantly that genes that are far apart on the same chromosome will appear to segregate independently in crosses. Okay, so there's a nomenclature that goes with this. VG plus means that the gene is wild type. If you just write VG, it means it's mutant. So B might be black body or it might be brown body, but it means that B plus means that it's a normal color and B is a mutant phenotype. So the plus symbol is used to denote the normal or wild type phenotype, and just a letter or series of letters denotes the mutant form. You don't use the big A, little a nomenclature in fly, but if you want to, you can. Okay, so here's an example of a cross between a Drosophila cross, which is really more like what they give you when you are doing these things. You had a fly that was heterozygous for the autosomal recessive, vestigial and black, was test-crossed to a recessive, and the following progeny were obtained. Well, you've got 400 vestigial, 400 black, 100 black vestigial, and 100 wild type. So the first thing you have to realize is they're not giving you all the phenotypes here. If the fly has vestigial wings and the body color is not mentioned, we assume a wild type. So this is actually VGB+. Similarly, the black body, the wing phenotype is not mentioned. So we assume it's VG+. Okay, the VGB is double mutant and wild type is wild type. So we could calculate the map distance fairly easily from this because we can see that the less prevalent phenotypes are the wild type and the double mutant. So those must be the recombinants, and the other two must be the parentals. So we just say 100 plus 100 over the total is 1,000 gives us 200 divided by 1,000, which is 0.2, which is 20 map units. Okay, so you notice that these are the most prevalent. So you have to remember that the fly has a phenotype for its body and its wing, and you have to write those in. And it's always a good idea to keep them in the same order. So here's my own way of writing this, get the same answer. If you really can't stand the pluses, you can use a big B, little v, big B for this. It really doesn't matter. But you have to make sure that they're legibly different from one another so you know which allele is which. Okay, so this is confusing. When you're writing these, when you write the parental configuration, you look at the parentals and you know which are the most prevalent. So in this case, the vestigial and wild-type body are the one parental, and the normal wing and the black body are the other parental. And so you write them with the horizontal bar between them. And you can do the same thing with the little v and the little b, whatever you like. So here's my map distance. 100 plus 100 divided by 1,000 is 0.2. Multiplied by 100 is 20 map units.